So today, my goal is to sort of set the stage on what is QPCR, why do we use it, what do we want to use it for, and then sort of after describing the nuts and bolts of how QPCR works, I'll introduce the topic of digital PCR. It's new, it's exciting. It's not the greatest tool for everything, so that's going to be, besides just the introduction, sort of introducing when you want to use QPCR, when you want to use digital PCR, because it's really, I fell into this trap too. I love instrumentation. It was always the thing that I got the most excited about. I got to learn the new thing and use the new tool, but it's not necessarily the best for everything. So always picking the right tool for the application. So that's going to be the focus there. But we are going to start with the building blocks, which is talking about nucleic acids. When we're working in qPCR, we work with two different types of molecules. We have DNA and we have RNA, just highlighting those differences. We have DNA, or sometimes talked about as cDNA, so deoxyribonucleic acid. It's really, when we're talking about cellular DNA, it's the information center. It's the storage of information. It's passed on the whole to other cells. There's usually just one copy per cell if you're healthy. Those come in different bases: adenine, guanine, thymine, and cytosine. So A, T, C, and G. Usually nucleic DNA is double-stranded, and it has a very long shelf life. You can do a lot of really aggressive techniques and your DNA remains intact and of good quality. Conversely, we have a different molecule called RNA, so that's ribonucleic acid, and it is really the signaler from DNA out to the rest of the cell. So DNA will actually copy for RNA, so that signaling is really important to a lot of cellular functions. It sort of, because it's the signaler, how much of it you make, when you make it, what is the sequence of it, all have an impact on what happens downstream. So it's positioned as a very important molecule. So in contrast to DNA, we have adenine, uracil, thymine, and cytosine. It's typically single-stranded, not always, but typically. And because it has a different molecular structure, we don't have the deoxyribose component, we have just the ribose component, it is less stable. It's more prone to degradation, which is a difficult thing for us in the lab because it's less stable. We have to do a lot of techniques to make sure it doesn't degrade. It stays of high quality. It stays the same amounts that we started with. But actually, biologically, that's very important because we don't want the on-go signal to remain on forever. It needs to go away and turn off after it finishes its process. So it's actually important biologically that it's made that way. It just makes our lives as researchers slightly more difficult. So just a picture, when we talk about base pairing, this example is of DNA. And what I want to point out is that what we're talking about when we talk about base pairing is that these molecules are actually hydrogen bonding with each other. So we have this, let me get my pointer out. We have these associations with each other. With adenine and thymine, we have 2 hydrogen bonds. With cytosine and guanine, or cytosine and uracil, we have 3. So if we're looking at how tightly everything matches each other on our 2 DNA molecules, we're going to have a tighter association with cytosine and guanine versus adenine and thymine. On what we call the backbone, that's where the key difference is, again, in DNA versus RNA. We actually have a phosphate molecule and our sugar ring here. That's that 5-member sugar ring. And that is where this attachment point, this backbone, is different between DNA and RNA. So I like this picture. It's nice. It's not mine. Thanks, NIH. So when we work with DNA, there are times where we want to measure what's going on and understand what's going on. And logically in this process, we ultimately want to get to the point where we understand QPCR. But to understand QPCR, we kind of have to make a stopover at PCR. So PCR is polymerase chain reaction. It's been around since about the 70s. It was a lot more difficult to do back then. We can all be happy we're doing it today and not 50 years ago. But essentially what we're doing is we're looking at a region of interest in a molecule, DNA or RNA, and we amplify that region for further analysis and study. So it's that copying. We can make millions and millions of copies of this target region and do our investigations. We can send it for sequencing, we can measure how much we made, and we can do a lot of things that way. What are the reaction components of PCR? Well, PCR has primers, and those are 2 starters. I've got some pictures for this, but they're little 25 base pair starter molecules. And because we can define where those are and make a specific sequence, we can amplify specific things in our target. Then we've got target DNA. This is our starting material for our amplification process. It's of your choice. I go out into the parking lot or the bathroom and I collect swabs off of everything and see how dirty it is. That's sample. It can also be material from a cell line. It can be so many things. There is DNA everywhere. And you'll understand that when you start working with these amplification reactions. There's DNA everywhere in life. So those 2 points are specific to each and every experiment, and you get to pick that. The more universal pieces are dNTPs, and those are the individual singular nucleotides that we're going to build onto the primer to create that sequence we're interested in. So they're nucleotide triphosphates, and they're usually in a mix. And it, like I said, it's the building blocks there. Then we have the enzyme. So we can't just throw everything in a tube and cross our fingers and hope it all builds the way we want to. We have to give it direction. And we do that with that enzyme. The enzyme can be used over and over in this reaction, but it is what really does the work. And then overall, we need to make that enzyme happy so that it works effectively and properly. And we do that by adding salts and buffer, creating our happy environment. Making our lives even a little bit easier, we can combine and buy these components from where we'll make them, and they're often sold in a 2x master mix, which means half of your reaction is these components in already appropriate concentration. So again, we're making our lives really easy. So now all you have to do is go get a bottle of master mix, add your primers and your DNA, and you're making PCR. So what's actually happening? So this is my nice little picture of what's actually going on in our reaction. So we have our template strand in blue. So imagining if we have DNA and we pull it apart, we're just looking at one of those strands. When we have the primer in solution at our appropriate temperature, that will bind here, and that's in yellow. And then the enzyme will bind in front of that and start building. So whatever's in front of it, it matches the next base, and it moves to the next step, and it just keeps going, and it builds and builds and builds. If you imagine what's actually going on in the reaction, so we can go through one process and build once. We copied once. The beauty of this with our enzyme and this temperature control is we can do that over and over. So what we do is after our first copying session, we had double-stranded DNA. We peel those apart just by increasing the temperature, and they melt apart. Then we lower the temperature, bind our primers, build, and then we can do that again. Raise the temperature from our now double-stranded after our first amplification. We raise the temperature, peel them apart, and copy again. So we're going to build on that twofold every time. This is very effective at making a lot of material, and it grows very, very quickly. So if we talk about running this little math piece here, if we have 30 amplification cycles and we start with one molecule when we start, after 30 of these reactions, this duplication, we will have 10 to the 8th copies. So it grows rapidly. So what do these amplification reactions look like? Typically, we have a step at the front where we activate our enzyme. So to make sure that our master mix works for you, we put molecules around the outside of that enzyme as protection. So we start this process during our activation step and peel all that apart, and now the enzyme is ready to work for you. Then we go through high temperature, low temperature cycling. That's our keyword. Typically 40 times. If we're designing our own primers, we're going to want to hit something in the 58 to 62 range or so for a melting temperature for those primers, and that's where we're going to extend. So cycling is the high, low, high, low. And then if we're doing just PCR, you have the option of finishing us all out at a 4-degree fold, because a lot of times when you do PCR, you're going to collect that material and do something else with it. All right, what can you do with it? So a lot of times people will run this on a gel, an agarose gel or something similar, and you can get an idea of what you made based on size. And if we migrate this in a gel using our energy field, we can compare it to a ladder, which is here on the far right of the screen, and that gives you an idea of what's the size of our product. It's an estimation, but you can get pretty accurate. You can also see, do I have one band or do I have two? Essentially, did I make one product or did I make two products? Other things you can do, you can estimate quantity on a gel, but if you want quantity, it's probably better to look at UV absorption or a nanodrop or something similar. But the drawback here is that all the products are going to produce signal, whether it's the product you're interested in looking at or something else you accidentally made. It's kind of an all-encompassing result. So that leads us to the next question, which is, well, how do I quantify a specific result? Yeah. Go ahead. Okay, so in this experiment, I just pulled this as an example. It's not intended to show you anything. Units are the amount of polymerase enzyme that was added to this reaction. So this experiment is looking at how much product you made versus how much enzyme you added, which really isn't applicable for our talk, but I wanted a nice picture of a gel. Units. Yeah, and that's how we typically talk about a quantity of an enzyme is a unit. Yeah. Okay, yeah. Yeah, no, you're going down a rabbit hole. So I'm going to touch on the rabbit hole and then bounce back out because I don't want to fall in. If we look at, say, we're all humans. Let's talk about the human genome. There is a lot of genes that are represented as homologs. And if we start going back in lineage, this goes all the way back to individual cells. There's a lot of genes that are very similar and do very similar processes. So when you are designing primers, there is a lot of work you have to do to make sure you're actually making what you think you're making. There's also variation in that melting temperature that we choose. If things are too similar, you can make a little bit of something else. So we have to be really careful when we go through this design process and developing an experiment. A lot of people talk about, oh, I just throw in some primers and it works great. But if we actually do this well, there's a validation that happens up front that makes sure we only made one thing. And we made exactly what we wanted to. And it's not trivial. If you do it properly, you can do it well. And there are some regions that are easy to design in, and there are some regions that are terrible to design in. But if you don't look for it, you're never going to know. And this is the cautionary tale of that. Just because you make something and it looks great on a gel, that's not necessarily your end of investigation. Yeah. Typically, 40 cycles is what's recommended, but that's for qPCR. For regular PCR, it's enough to make what you need. So it's a bit of it is a different technique and it has different bounds. If we're talking about qPCR, our default is always going to be 40 cycles. All right, so the question then becomes, well, I want to make a certain thing and measure it. Because remember when we talked about RNA, how much we had and what we had were both very important to the signals it sends and the information that's being transferred in the downstream processes. So how do we do that? Well, this is a nice list of a lot of the different kinds of applications besides gene expression that we can do. So today I'm talking about gene expression. That is usually the most common application people see, but just want to highlight there are a lot of other things you can do with this technology. So let's get into it. So the three most common, again, we have absolute quantification. That's going to be taking a known, diluting out, say, a standard curve, comparing an unknown to the knowns, and back calculating how much you started with. Gene expression goes by several different names. So I want to make sure you hear them all so that you can navigate your way through this space. We have comparative CT, we have relative quantification, we have delta-delta CT, we have gene expression, we have fold change. These are all words that are part of gene expression. So I just want to make that clear. Can you use this for? Gene regulation studies, disease mechanism, therapeutic targets. This is a big bucket. Other things you can do with qPCR, the third most common that I see is pathogen detection. See that a lot in, we saw it a whole lot a few years ago. So all the PCR testing, when people talk about PCR testing, we were using qPCR. That is a non-quantitative, do I have it, do I not technique. Chris and I have been having a conversation recently about looking for environmental DNA and how much of that do I have. But you have to know if you have it before you move forward, and that would be detection. If I go outside to say we're in Dairyland. If I go to a dairy farm and want to make sure that the cattle is well, or there's a virus going around and I want to make sure they're not sick, I can do sampling around there, test for those targets, and I can put the farmer's mind at ease knowing that his herd is healthy and is not likely to get sick, and we don't need to call the vet. So we can save money, we can be proactive with the health of the animals, lots and lots of different things. All right, so these are some of the things we can do. Again, we're going to be focusing on gene expression. Yeah. You could do it faster in QPCR because you're simultaneously, we're almost there. You're simultaneously making and detecting. Whereas when you do QPCR, you have to make, and then when you're done, you analyze. So that's the difference. So yeah, go ahead. Yeah, go ahead. No. Yes and no, and we're coming up to that. But I'll answer the question, and then we'll revisit it when it comes up on slides. RT does stand for reverse transcription. Okay, so RT-qPCR takes the reverse transcription step and the qPCR step and does them together. So it's two actions at the same time. So that's the nomenclature difference. We'll talk about what is reverse transcription in just a second. So how is mRNA or gene expression, these signaling molecules, positioned in the bigger picture? We have DNA that is transcribed into RNA. RNA is then translated into proteins. So if you have a protein, you can actually somewhat backtrack your way to what you started with, or look at your DNA sequence and say, oh, this is the RNA I'm going to make. And then take that RNA and say, like, oh, this is the protein I'm going to make. So if there's errors, you can see that differences, say, in the DNA could work its all the way down to a different amino acid. So that's how that can kind of change. So the amount of that RNA we have can change how much of that protein is made. Proteins do actions. And then if the protein, like I mentioned, if the RNA is different, it can lead to different amino acids, which means you have a potentially different conformation of your protein, which can do a different thing or do it in a different way or not do it very well at all. All right, so again, we had that twofold amplification process, which is represented again slightly differently on the left. Sorry. That's the right. And the difference between PCR and qPCR is up here at the top. We do the cycle. We take one molecule, we make two molecules. We then measure that between every cycle, and we count how much did I make. We do a cycle, how much did I make? We plot that fluorescence on a graph, and we can watch that exponential growth, twofold growth, exponential, biology, we love those. So it starts out really slow, we're making some, but we can't detect it, and then it takes off when we make a lot. When that takeoff happens is the information we use for our quantification techniques. We call it a CT. I'll come back to it. There are two different big camps on how you can actually produce this fluorescence that we can measure inside the instrument. You can do it with cyber. So this is the next intellectual progression in what we've already talked about. We have two primers. We amplify. We use this double-stranded binding dye called cyber that sticks to anything that's double-stranded. We get signal from that. We shine a light on it. We measure how much signal we had. We record that. We do another cycle. We made a bunch more stuff. We put light in. We measure it, and so on. This is how we can generate that data. Unfortunately, you had a great question in the back earlier about specificity. Cyber is still a non-specific binding molecule. It sticks to anything that's double-stranded. So if your primers stick to each other, you're going to get signal from that. If your primers make the wrong thing, you're going to get signal from that. So as much as a lot of people in academics, and you're going to get pressure from other people like, oh, I use cyber and it's great. It can be. It can also potentially lead you to an incorrect result, which is why I caution people, if you're going to do cyber, do your background work. Make sure you're making what you think you are. Okay, it's not a guarantee. The other camp. So I cautioned you against cyber. What's the other camp? The other camp is what we call TACMAN assays. And that really is what, back in the day, applied biosystems really was one of the big cornerstones of our whole company. And that was developing tests using this TACMAN technology. So what does it mean? We have the same two kinds of primers that start amplification. But in the middle, rather than getting our signal from this non-specific cyber reporter, we're going to get signal from a sequence-specific molecule we call a probe. So a probe is going to have a fluorophore on one end and something to quench that fluorescence on the other. And if it's floating around free in solution, those two interact and you get no signal. If you get amplification and that enzyme runs through that probe, it's going to get cut apart. And now you have a fluorophore separate from your quencher, and you're going to see fluorescence. So there's a lot more specific things that need to happen in a TACMAN assay before you even get to the point of signal generation. So it tends to give you a more specific signal. You're just starting out. How do we do this? I mentioned we have master mixes that can, they're pre-formulated, ready to use. If you, again, have questions about which ones to use, you can use this link and go through a questionnaire, and we will try to present you with a master mix that's going to work for you. Otherwise, call for help. You know, our website is gigantic. I understand that. I get overwhelmed trying to navigate our website. Sometimes it's best to just call somebody. Our technical support team, this is something they do on a regular basis. Just call or email. They're going to be able to help you out. They're ready and able to sit there and answer your questions. From here, we have either a cyber master mix where all you need to do is add sample and primers. We also have TACMAN master mixes where you add your sample, and we, as I mentioned, we have pre-designed assays. So we have already done the work of making primers and probes in a set to do what you want. So if you go to our website, which is up in that link, you can browse what we already have. We have over 20 million already prepared assays ready for you to use. If I was in your shoes, that sounds really awesome that somebody's actually looked at and made and worked with assays, and I don't have to do anything. Yeah. Yeah. I didn't understand your question. No. So I think you're trying to combine like Sanger sequencing with QPCR, and no, they work a little differently because it's about getting fluorescence without background fluorescence. And we do that spatially in Sanger sequencing, that we have a molecule and we look for fluorescence of a nucleotide, and we separate that in space by passing it past our detector in a electromagnetic, in a charged field. If we tried to do that in QPCR, we would see incorporated and unincorporated nucleotides at the same time, and we wouldn't be able to tell the difference. Yeah. All right, so our assays. I used to love to do assay design. Like, it was my jam. Loved it, it was awesome. I was really good at it. I spent a lot of time doing it. I made the switch to ordering my tests, and I got sad. Not because they didn't work, but because they did work. And all of that lab time, I got to design experiments and look at the minutia of, optimization and trying to, validate that I made what I wanted to make, went away. All that work just evaporated. So I highly recommend this. I was sold on these pre-designed assays before I was a customer here at Thermo, or as a customer before I started on at Thermo. Again, the website can be really confusing. So if you are not confident on what you're looking at in this search tool, let's talk. But one thing I do want to point out, it's not evident when you first look, but we have something called the extra small size. The extra small size will get you 75 20 microliter reactions or 150 10 microliter reactions. It is a very low cost, high success tool. So I highly recommend that you invest in these assays. When it comes down to it, when you start thinking about time and expense to do the proper cyber or PCR validation, making sure you're making what you want to versus just getting your answer, it's usually worth it. So look at that. And Lenny can help on pricing, so you can talk to him. Yeah. I'm not supposed to, but I will. If you look at our website today, the catalog price for everybody without a discount is, I think, less than $110 for a complete assay. And that is enough to do 150 10 microliter reactions. So for most people, that's a whole study, at least for you guys. That's the high point. It should go down from there. The university gets great pricing. So it should be much, much less than that. But Lenny's the one to talk to. Yeah. Yeah. So let's talk about the data we actually generate. It's actually the next slide. So when we generate data, what are we actually looking for? So again, we talked about growing that twofold target up in quantity. And again, this looks like flat background. It's growing, but this is our minimum detection, so it stays flat. It looks flat. You're growing, it's just not detectable. And then you get to the point where it really takes off, and this is the point where we want to do DNA quantification. And then at some point, after you make a ton of stuff, oh, sorry, I've got one. Okay, thanks. I'll stand here. Okay, so we've got our baseline, and then we take off into our exponential growth phase. This is where we can actually see that twofold growth, right? We see it. We have twice as much as the cycle before, and we can measure that. That's the high accuracy region, and so this is where we look to do that quantification. We then move forward into what we call the linear phase, which is where we're still making more, but we're not making twice as much every cycle. We're making a little bit less. So the question is, why? Well, usually it means that we're starting to run out of something. We're starting to run out of one of the reagents in our reaction. What can that be? That can be our NTPs. It's usually our enzyme, or not our enzyme, our primers. And we can actually utilize that for some different techniques. But at some point, you're going to completely run out of something in your reaction. And that's where we get to what we call plateau phase. So you've completely exhausted what's in the reaction, and you can't make any more fluorescence because we've run out of something. So you can run it over and over and over, and it's just going to run flat. Okay, that's the plateau phase. So just to highlight here, plateau phase doesn't have anything to do with your sample. It has to do with your reaction components, independent of sample. So just because you see a difference in your plateau phase doesn't necessarily mean that you have a difference in quantity of your sample. When we actually do our data collection, because it's really hard to look at this point and say, what's the right place to put this threshold? Where do I actually want to gather that data? This is on a linear scale, so it's hard to see here, but remember, this is a twofold growth process. So if we plot it on a log scale, we can expand that region of high twofold growth. So when we look at it on a log scale, what we're looking for is this nice flat portion of the graph, right? If we have a twofold growth process, plot it on a log scale, it becomes linear. Okay. And again, we get to a plateau phase out here. So what is, I've said the word before, but what is this threshold? So we're trying to quantify how much we made, but we need a set point to collect that data. So for all of these different samples that we have, each one of these groups is a different concentration here. We need to set where we're going to collect that data and be consistent for the whole experiment. We do that by setting this threshold. So we set a threshold line that goes through that flat part of the amplification curves above the variability of background and below where we run out of twofold growth in that linear phase. So when it rolls over, you know, you're out of it. So anywhere in here is a good place to collect that data. And I circled it. So what is CT? CT is the point where the amplification trace you've made crosses the threshold line, and then we can get this fractional CT value that we can use for our data analysis. So each one of these groups has a different CT value, and how much they change determines the amount of difference between those two groups. What does the CT actually mean? Okay, the term definition is the cycle at threshold. Okay, where does it cross that point? Sometimes you'll see a term called CQ. It is cycle for quantitation. The way we're talking about it today, they're the same thing. So mentally, you can kind of swap them out. If I move my threshold, which I did here in these two images, I moved my threshold up. So I changed my CTs. They were 25, 26, and 27. I moved that threshold to make them 26, 27, 28. Did I change how much DNA I made? No, I didn't. This is the same data. I made the same thing. But where I'm collecting the data is a little bit different. However, the difference between these samples remains consistent. Between 25 and 26 is 1 unit. Between, on the left, 25, 26, 1 unit, 26, 27, 1 unit. CTs are all relative differences. They have no absolute independent meaning. They only have meaning when you compare it to something else. Okay, that is really critical. Okay, you can't just walk up and do an experiment and say, well, it was 25 today and it's 26 the day before, so clearly that's different. That's not the same thing. If they're on different plates, run at different times with slightly different analyses, that means absolutely nothing. If they're on the same plate, run at the same time. Now you have meaning because you're comparing. Yeah. Keep going. You can see both. Typically, the default view is going to be the log view because we want to use it for quantification. So we're typically going to present that to you first. But you can see both. Log view. Yes. Yeah, good question. That's an excellent question. So where do you set the threshold? First of all, the default in the software is that we have an automatic threshold setting. So the software tells you, this is a great place to put a threshold. I'm just going to set it here, and you can change it if you need to. So that's starting point number one. It will do it for you. After that, if it doesn't look right and you want to change it, you need to have your threshold above the variability of your background, which is here. This is a log scale. So this really represents a small amount of material, okay? A little bit of amplification. So you want your threshold above the variability of the assay. So you can see here, this is pretty variable. It's not real consistent. That's not great data. But we get really consistent here in that flat region, again, on the log scale. And you want to keep it in the region where this is flat, okay? This region is flat. Once it starts to curve over, you're out of the exponential phase and you don't want to quantify there. So we can draw a line at the top about here and the bottom about here. Okay? Yeah. Sorry, it's what? Yes. Yes. It's one reaction tube. Typically, when we run gene expression, we want to have 3 runs of exactly the same thing. And that allows us to do some averaging, outlier elimination. So if you think maybe one of them is bad, you can throw it out and you still have good data. Or you have an average, which means you get error bars. Error bars are great. People shy away from them, but statistical confidence. All right, so we talked about CTs. Let's talk specifically about gene expression. We've collected a bunch of CT values. What do we do with them? All right, so again, just some descriptions of what you can do with it. We can collect from several different groups of people and evaluate how those different people, those different groups have different expression levels. I've sort of outlined a general experiment. So when we do gene expression, first we need to collect our RNA sample. Okay, we collect that, and then we have to do reverse transcription. We had a question about what is reverse transcription. So let's get into it. So we can't effectively, the enzymes we use to do the copying don't work on RNA very well. Okay, that's point number 1. Point number 2, RNA is really unstable, and we're asking it to do these high temperature cycles. What do you think is going to happen to RNA if you put it in a tube and let it sit at 95 degrees C? Not going to have a whole lot of RNA for very long. We're trying to be quantitative about this. Every molecule makes a difference. So in order to start this off correctly, we are going to take a first pass and we're going to make a 1 copy pass and turn our RNA into cDNA. C as a copy, okay, it's copied DNA. And we can do that independently using, we have prepared mixes. So if you want, there's basically 2 camps of this. Again, we have lots of options in science, but really we want to start and say, how do we want to do this? You can do it in 2 steps and say, I've got RNA, I'll make cDNA, I'll put it away, and I'll do the rest of my experiments when I'm ready. That's 2 steps, right? RT, QPCR. The other option is to do them together and do RT-qPCR. And what that means is that we have a master mix that has both of those enzymes, the enzyme that can do the reverse transcription and the enzyme that can do the cycling amplification process in the same tube, okay? Advantages of either one. Well, if you don't have all your material at the beginning, you can't go ahead and plow forward with data collection. Or if you want to keep that material around for a long time and archive it, you know, you're going to have a much more stable cDNA than trying to hang on to RNA for a long period of time. Okay? So if you're doing a longitudinal study, that's a great place to use a two-step. If I do two separate reactions, I'm now pipetting a bunch of things. I'm now doing that twice. You introduce some error with that. How good are your pipettes? How good are you at pipetting? It is a skill, okay? You need to get good at it. So to start off, it can be good to start with a one-step just because all of that happens in one tube. It's not always the right choice. Yeah. Okay, so when you do the RT reaction, the mix, this reverse transcription will have all of the components to do the copying. It has the enzyme, it has the material to do that. In addition, you have all the other components. So if you're doing a one-step, everything sits in one pot, and we separate those actions based on temperature. You do the RT step at 50 degrees. Once you're done with that, you can then go forward with your enzyme activation for qPCR, and you move forward with amplification. So we separate the actions by temperature. So we have our qPCR enzyme that doesn't start working until we get to 95. So we start the whole thing at 50 and do RT, and then go on and do qPCR. Make sense? You can. Yeah, in one step, you do them both end to end. Okay, great. So in our mock experiment, I wanted to look at certain expression levels. I did my RNA isolation. I'm going to do one step because that's easy. All right. So I do that reverse transcription. I do my qPCR amplification. That's my reverse transcription. Then I do amplification in qPCR. So what things do I need to measure to actually be able to do the calculation and understand gene expression? We need two, this is the bare minimum. We need two kinds of samples. One that I'm going to compare everything to, and one that I want to understand. So control and reference, untreated versus treated. Time course is going to be time 0 versus my other time points down the road. We need two tests. So now we've got two genes. So a gene of interest and a gene that we know doesn't change. Okay. Sometimes that's going to be called your endogenous control or your reference gene. But again, we need to compare something that's not changing to something that's changing. Okay, so those are the four basic key components to gene expression calculation. Okay. How do you pick that? Well, we have some heavy hitters on that list. You're going to see these a lot. Essentially, you know, ultimately, definitively, which is the right one to pick? It depends on your experiment. How is it treated? If you're doing a time course and you treat your cells, well, you have to test. Does this change or not? Okay. You can trust other people and pull from publications. That's fine too. You know, I have had it happen enough that what I'm doing is slightly different from what previous researchers were doing. My process is slightly different, and I have trouble trusting implicitly what's in papers, personally. You're an experimenter. You get to choose those. All right. So I had a time course. Again, I mentioned I've got RNA I'm collecting. I am going to go ahead in my amplification reaction and do RT-qPCR. This is my result. The green is my gene of interest, my target gene, the one I'm interested in looking at. So we had the four components, and they're represented here. Target gene in green, endogenous control in red. So for every sample I have, I'm going to compare those two. And then I have my time 0, 12 hours, 24, and 48. Okay, so those are the metrics of 4. 0 versus unknown, target gene, control gene. All right, and then we can tabulate the data. Great, tabulating data. But again, remember, all of our calculations are based on differences. So we got to start doing some delta calculations. Remember, that was one of the names that we gave this, was a delta-delta CT. And that's what we're going to do with our data analysis. We're going to generate not one difference, but 2 differences. So we call it delta-delta CT, or comparative CT, because we're comparing CTs. I know, right? Okay, so first thing we do is our delta-delta CT. Oh, I hid too much. Sorry, I'm going to undo that. We will undo. All right. All right, so first things first. We have our first delta, where we calculate the difference between our gene of interest and our endogenous control. So column 1 minus column 2. Then we have our second delta calculation, which is going to be the difference between samples. Okay, so we have row 1 compared to row 2, row 1 compared to row 3, and row 1 compared to row 4. Okay, and these are straight up subtractions at this point. Yeah. Yep. First delta, remember it's delta-delta CT. First delta is comparing the 2 different genes, and we tabulate that in the single delta column. From there, we compare samples to each other. That's the second delta. So it's line 1 minus line 1 is 0. Line 1 minus line 2 in the single delta. So 11 minus 9, negative 2, 11 minus 4, and so on. So that's where the data comes from. Right? That's the next column, RQ. So RQ stands for relative change or fold change. So by definition, our 0 point compared to the 0 point has a value of 1. It doesn't change. That's what 1 means. If we look at the 24-hour mark and compared that fold change, the RQ was 4. So we had a 4-fold increase. What is that calculation, which I think is the piece that you were missing, is this. So we take that delta-delta CT number that we generated in the second to last column, we plug it into this equation because this is a 2-fold growth process that we need to undo to actually get numerical values and a linear understanding of the numbers. Once we do that, we get that RQ value in the final column. Sorry, that should have been in the middle. So the meaning of RQ, 1, no change. A positive value for RQ is an increase in a fold. So 4 means I had 4 times as much in the 12-hour sample as I did in the 0 time point. 256 for the 24-hour sample means I had 256 times the amount of material for this target in the 24-hour sample than I did when I started at time 0. After we get another 24 hours in the future at 48 hours, we now have a 0.25 fold change. meaning we had just 1/4 of that original expression level at time 0 that is now reflected at the 48-hour mark. Okay, so that is how we interpret fold change and how we calculate fold change. Good news. Going through that more specifically. If you do it inside our software, it does it for you. If you assign all the control genes, all of the target gene, the control gene, the reference gene, or the reference sample, all of this is calculated for you. Okay, so all you need to do is grab RQ and plot it. It will plot it for you, but not everybody likes these plots. So if you like different software, you can pull it out. It also does the really hard part of giving you error bars. So we have RQ min and RQ max, meaning the lower bound and upper bound for your error bars. Just a reminder, this is a twofold growth process. So your error bars are going to be asymmetrical. Okay. It also means trying to calculate this in Excel trying to execute this in Excel correctly can be difficult, okay, because it's logarithmic. Yes? Yeah. Remember the way we are evaluating our data. Yeah, this is a good point. I'm glad you asked that. Let's get back to a data slide. Let's look at this one. If we think about how we're generating data, if you have a lot of material, you're going to see it earlier, right? Because you've made enough to cross the background level and be able to see it. So actually, more material comes from smaller CT values. It's a little counterintuitive to everything else that we measure in the lab, and why in our table, it looks like the smaller number had more stuff, but the smaller number does have more stuff. That's how it's defined. And one of the mental hurdles we have to go through when we learn this technology is that everything we think we know is kind of turned upside down. So we have to really think about what we're doing and how we're talking about changes to make sure we're talking about them in the correct direction. When you're doing your data analysis, negative values, positive values, and sign changes can make a huge difference. Okay, so you do need to be careful. Excellent question, thank you. All right, so we made it all the way down here. Anything else before I go? Oh, not the beginning. I got it. We do it. Okay. Anything else before we move forward from data analysis? And this afternoon, we'll go through how to do that in the software so you can get some hands-on with that. You can see how it's done. All right. Yeah, go ahead. Yeah, so that depends on how long it's going to be before you use it, honestly. Things that can degrade cDNA. Long-term storage at different temperatures mean it's going to degrade at different rates. But also things that degrade cDNA are low temperature storage and freeze-thaw cycling. So if you're going to use that cDNA tomorrow, I'm going to leave it in the fridge. If I'm going to use it, or maybe I'll use it some point, I'm going to put it in the freezer, -20 or -80, depending on when I'm going to come back to it and how deep into the storage archives this needs to be placed. So again, not a direct answer. Don't you love that? I'm giving you non-answers. it's cDNA, so it's really stable. It's not RNA anymore. You don't have to like grab onto it and make sure it's good. You can just let it freeze. That's usually good, yeah. Yes, you can. Again, you need to be really careful about how you're handling that. So every freeze thaw, and you can prove this to yourself. It's something that I did because I was like, what really happened? So I took a sample, put it through 3 freeze thaws, and tested it at each of those time points, and you can see its quality and quantity degrade over time. Yeah. So typically you want to get to RNA as soon as you can. If you're talking about something from, yes, there's some nomenclature within the individual sentence that you meant that can mean different things depending on what you're actually trying to accomplish. So remember, our end goal in our gene expression experiments is fold change. Well, if I only put in 1/10 of a material to start with, that's going to impact what my CT is. But remember, the end point of this is looking at how it compares to others, how it compares to itself more than how it compares to other samples. So that fold change can actually be really big, but I only put in a little bit of stuff to start with. Fold change is going to be the same whether I put in 10x, 1x, or 0.1x, but where those values show up is going to change in the graph. Does that make sense? Okay. Yes, at the right point, right? Go ahead. Yep. I'm sorry, I just got lost. Good. For gene expression, we need to remember we had 4 specific components. We need a sample to compare to other samples, but we also have this, we need to understand how much we have. Maybe that's a good way of talking about it. Our controlled, right. Yeah. I think you can do it that way. I think you're doing it the more complicated way. I'm not going to say you're doing it incorrectly. I think you have an understanding of what you're doing. I'm moving when that happens. Yeah. We have a fundamentals guide, not fundamentals, what do we call it? The handbook. We have a QPCR handbook outside that is a great reference. It's also available online if you want to have this, and it goes into a lot more detail about how to collect and do the RQ data analysis. And I think that would be good for you to sit with and understand kind of where I'm coming from. But I think you're doing it the right way. You're just doing that transformation at a different point. But I think we're getting to the same end result. Okay, So some best practices. When we get into the lab, what do you actually want to make? Oh, sorry. That's another good point to bring up. If you are doing a one-step where you have one tube and you do your RT and QPCR in that tube at one time, no, you cannot go back in your cDNA. If you want to do another experiment, you're going to need to do the two-step where you do your RT, you have a tube, you put that away, you use some of it for this experiment, you come back and use some more of it for a different experiment. That's a really good point. You have choices. Sometimes some are better than the other. It depends on what you are doing. And if you need to come back to it at a later date, then you want to have a cDNA library and you need to do a two-step. Yep. Okay. So when we are working with qPCR, PCR, RT, RT-qPCR, we want to make sure that our DNA and RNA is of high quality and clean. So one metric that we can use for making sure that we have just DNA is an A260, A280 evaluation in a nanodrop. We can also take our samples through nucleic acid purification, spin columns, molecular spin columns, or magnetic particles, or just a good old-fashioned organic extraction. So good things. If you treat with DNAs around where you're working with RNA samples, you have a higher chance of reducing your RNA degradation, so consider that. It's nice to know how much RNA you're putting into your reactions, because if you put in too much, you can overwhelm the reaction and overwhelm your enzyme. So it's good to know how much goes into those before you start, and making sure you have a high quality reverse transcription reaction. Using our kits is a really, really good option there. Everybody uses them. Not everybody, but they're highly used. They're really good. Preparing your workspace. You know, it's nice to be organized and set it up appropriately and collect all your, like cooking, collect all your supplies before you start, and your whole process is going to go better. Yeah. Yeah. It's best practice. You don't always have that option, especially when you're doing a one-step. Sometimes you've got what you got. You know, you have an FFP sample, you put it in a tube, and sometimes you got a little too much. So it's good to do it. It is not absolutely necessary. We are not using it as part of the calculations, but it is recommended if you have that flexibility and time. Yeah. Typically, the more you can standardize it and check and double check, the better you're going to be, because it's really easy to make pipetting mistakes, and it's always easier to check and be correct and get the wrong conclusion. That's my worst nightmare. No, that's going to be high quality. Yeah. No. You're talking what the last slide, mostly what I was talking about was making sure that you had good material that goes into the reaction. So once you're in past that first hurdle of reaction, you're pretty much good to go. Yeah. Okay. So if you've never worked with these amplification reactions before, you're going to need to know that DNA is going to float around. It is like magical. It will float. Sometimes just opening a tube and then swinging it over your negative controls is enough to get amplification in your negative controls. okay? Seriously, it's like magic. I didn't put that there. Where did it come from? So we need to be effective and think critically about what we're doing and how we're doing it. We need to start in a very clean space. Some people, because we have this environmental systems that can hang on to these molecules, sometimes working in a PCR hood is a good option if it's available. That way, it's a space that you can shine a light in and clean it and degrade all of the sample before we start. And then it's clean for the next person. Dedicated pipettes, that's an option, not always something you're gonna have here in these labs. Using filtered tips though. I always recommend using filtered tips because remember how pipettes work, we are moving air to pull up volume. If you don't put a barrier between your sample and the inside of that pipette, there is the chance that the air hangs on to some of those DNA molecules, and the next time you use it, you're putting it in the next space. So really like the filtered tips. All right, again, here's a nice table showing the difference between 1-step and 2-step. Some information about our plastics. Today we're going to be using the 384 well plates and 10 microliters of reaction this afternoon. Just remember, when you seal the top of the plates, we need to have an optically clear, remember we're shining light through the top of the reaction. So we want to have it optically clear and completely sealed, which is what you see in the 2 images at the top, is we're taking that paddle and making sure everything is completely sealed. You always want to make sure everything is vortexed and spun down before we start. What's going on with this vortexer? I've taken a glove and put it over the top. If you've ever had vortexers that have been around for a while, that head starts to shed the little black things, the little rubber pieces. That's really not good to have inside the instrument. So if you put your plate on there to vortex it after it's sealed, you can pick up the little black little balls, and we don't want to put that in the instrument. So if you're going to vortex a plate, I like to have this as it's a protection. We're just protecting the head from the plate. All right, questions about QPCR? Yeah. This afternoon. Yeah. Yes. Yeah. They're both effective. That's a deep question. They both work. We have optimized our RT reactions to work in different ways. So what we're actually using in which tube, we'll have to dig into. So, again, what are you doing? Yeah, we have to ask questions before we can answer them. Yeah. Yes. So it's a great point about experimental design. What I showed you about running 3 reactions of the same thing is what we call a technical replicate. And that's to ensure the number we measured is an accurate number. What you're talking about is establishing biological replication. Say having 2 or 3 samples of your different time points and having those run in technical repeats to give you assurance that your fold change is an accurate fold change. Okay, so we have two things. Technical replicates are going to give you numerical bounds around the confidence of what you measured from that sample. And then we have another measurement for sample to sample variability. So that's biological replication. They're both important. We've now gone from having one data point to needing 9 reactions. It grows fast when you start doing that. So think critically about what you really need. Think critically about what you have money for. You know, there's no one-size-fits-all solution to gene expression. It's all about what you're doing, what kind of confidence you need, and what kind of results you're expecting. Every situation is unique. Yeah. You should. Okay. I know a lot of our older friends are, you know, maybe somebody who you learned from in the past has said you don't need to vortex plates, or don't vortex your plates because this is an enzyme and you can inactivate your enzyme, or something like that. So I very much disagree with that. Our modern enzymes are very robust. And if you don't vortex, so what's the risk in not vortexing? So if you don't vortex, potentially what you're doing is setting up a gradient inside your reaction such that you don't have a homogeneous representation of reaction components, which means you can delay effective or observable amplification slightly. That's going to impact the data quality and data consistency you get, because the CT value is going to be slightly inaccurate. Not incredibly inaccurate, but slightly. So it takes about 3 cycles to, if you have a completely static plate that you just layer your reagents on top of, it takes about 3 cycles for the heating and cooling process to effectively mix that. So if a 3-cycle shift is okay with you, you can probably get away with it. If you're doing a presence absence, you know, maybe that's okay because you're not trying to quantify. But if you're trying to get numbers, I highly recommend mixing. So you're going to vortex and spin that plate down every time. You don't need to worry about harming your enzyme or doing damage to your reactions. Very robust. Forward. We're Wisconsin, right? Forward. All right. So we got just a couple minutes to introduce digital PCR. I know. Your brains are full. Give me just a minute. Okay. So what is digital PCR? It is a hot topic right now, isn't it? You're reading papers, it's exciting, it's our new tool. So we talked about gels. That was PCR in our endpoint quantification. That's where we started. We just spent a lot of time talking about our real time. Real time is where we can quantify what's going on. Digital is going to be where we pick up on sensitivity of qPCR. So we're not getting the data we need. We have too little sample. Our differences are too small. That is a great time to move to digital. All of these methods use the same or very similar bulk components. NTPs, enzyme, primers, probes, buffers, and salts. And actually, a lot of our reagents are cross-compatible. qPCR and digital PCR, those assays we went through in the catalog, a lot of times, most of the time, they'll run on both platforms, qPCR and digital PCR. So they should be thought of as complementary. qPCR is fast, effective, cheap. Digital PCR is more expensive, but it gives you access to data you can't get in qPCR. So yes, it's more expensive, but if you can answer your question definitively, is it worth it? There's always a trade-off in our experiments in science, and that's what we're looking at here. Okay? There's never going to be a replacement for the speed and cost of qPCR. Just want to make sure these should be a handoff, okay? All right. Where digital PCR tends to really shine is down here. When we lose the ability to quantify, this ends up being somewhere when we think we have CTs of like 36 to 40. If you do the math, because we all love math, right? If you do the math, if you have one copy of material and perfect two-fold growth, you will be able to get a CT of about 40. You'll be able to see that after 40 cycles, okay? But how good are you going to be at consistently grabbing one molecule and putting it in a qPCR reaction over and over again? The reality is not very good. You might grab it with your pipette tip, you might not. Okay, there is an inherent variability when we get down to really low concentrations. Okay, that is where digital really shines. So I listed some applications here and some advantages. Other than just low concentration samples, actually here in the BioGarage, worked with a customer that had bovine rumen samples. Rumen is notorious for having enzymatic inhibitors. Okay, if you have an inhibitor, that means there's something in the reaction that's going to make it not work at that two-fold growth. It's going to inhibit and suppress that amplification process, which means their qPCR was inaccurate or not present at all, depending on how much of that inhibitor was present. We were able to compare their qPCR results run on the digital system. The digital data matched their functional data. qPCR did not match their functional data. And that's because we have this ability to get over some of those inhibitors. Yeah, so downstairs in the BioGarage, we have an Absolute Q system by Thermo Fisher, and they have their own controlling software for that. Right. Yeah. Yeah. Okay. So anytime, another good point to use for digital, there is a technological limit that we have to qPCR, especially in gene expression. The example I gave you was anywhere from 0.25-fold to 256-fold. That limit in qPCR is about two-fold. Anything less than about 1.5 or two-fold difference, we just, and it's not that we don't make good instruments. It's just across the board, everybody's technology. That 1.5 to two-fold change difference is the limit because of our uncertainty in measuring CT values and measuring the data. Okay. If you have changes that appear to be consistent below that value, move to digital, because its error bars are so much smaller, you can assess those changes and get down to about 10% difference, okay, 1.1-fold. So we now have access to a new kind of data that we didn't have before from qPCR. So that's, again, how they sort of work together. How does this actually happen? So in digital, we made, so if we compare this to qPCR, we make a bulk reaction, we put it in a tube, we put that tube on our instrument, and we amplify it. In the digital system, we put a step in the middle, what we are calling compartmentalization. So we take that bulk reaction that we made, we push it into teeny tiny little reaction compartments. We're talking 500 picoliter size. I mean, absolutely incredibly small. And then hopefully we have about, on average, 1 molecule per microchamber. We then put that through, we do amplification, and we can count up how many times we see amplification and how many times we don't. From that number, we apply some statistics and we get a concentration value. It's as easy as that. It's a binary counting system. Digital, yes, no, on, off. That's what we're doing. We're counting yeses and nos. So good uses of the two technologies. Absolute quantification, a really small dynamic range. You don't have much variability, but it's consistent and important. Excellent precision. So if you're doing release of a drug or a biological drug, you need to know that it is safe for people to take into their bodies. This is an amazing solution for that. Have companies that do exactly that. Tolerating efficiency differences, remember those inhibitors, that's good too. And then it's good for the lower throughput end. We've got limitations for cost and time. Our QPCR instrument can run 384 samples at one time. Our digital system can run 16. So we have a big throughput difference. Real time, again, it's an amazing application. It's tried and true. Relative quantification, gene expression, bang on, do it. We have a huge dynamic range. We have something like 8 logs of dynamic range in QPCR compared to 4 to 5 from our Absolute Q digital system. Has good precision, not great, good, which for a lot of applications is all that you need. And then it has extremely high throughput. So again, what do you need to know? And what are you willing to, sacrifice for it? That's, again, what we put through here. Have some images. So it's a picture of the instrument and the consumables. I love this picture. You can ignore what it says at the top. This is a picture of the plate, and then we zoom in on it. So what we're doing is we're pushing, where our sample goes in here, it gets pushed by the system into the channels. It then gets pushed into these individual locations. We flush it with an isolation buffer, and that allows us to do our individual reactions. So the good news for all of these applications is we have our master mix prepared that you can buy from us. We have those assays. Bring your own, buy from us. Those are ready to go. Those assays can go in the digital system. They can go in the QPCR system. Master mixes, we have separate master mixes pre-formulated for the digital system. Buy a tube of that. We have a master mix for QPCR. You can buy that. You put all those together. You put them in a tube. Those tubes go on the instrument. In both cases, either the bulk reaction gets loaded here, or you put it in a plate and it goes on the QPCR system. From there, it's hands off. You don't have to do anything else except come back and collect your data. Okay? That's why I really like this digital system, is because it's really easy to get started. It's really easy to just throw spaghetti at the wall and see what sticks. I have had so much success here at the BioGarage doing that. I want to give you confidence that you have the skills to do this, or you will learn them very shortly. Okay, you can do this. All right, so we're at time. So I am going to save this for another time when we want to talk about a more in-depth discussion of digital PCR, but now is time for a break, and then we can go and watch our lab demo. All right, thanks everybody.
So today we are going to do a experimental, we're using a control kit, the TaqMan Gene Expression Control Kit and TaqMan Fast Advanced Master Mix to do an example of what a gene expression experiment is going to look like. So when we were in the conference room, we talked about master mix, and this is one of our pre-formulated, one of our favorite 2X master mixes. If you're going to be sitting for a long time, you want that to be kept on ice. So this is a 2X formulation. We're going to prepare 10 microliter reactions today. So half of that, 5 microliters, is going to be the 2X master mix. Our TaqMan Gene Expression Control Kit comes with 3 items, which I will open for you. That comes with 1 control sample, and 2 assays. So we have 18S and a TERT assay, both for human genomic DNA, or sorry, human RNA, and then we have our sample of the human RNA. So what I'm going to do is when I first start, I'm going to vortex everything that had been on ice, which were these 3
Turn it up. He's got the volume up as fast as it goes.
Okay.
Yeah, I'll... Wait. Do you want to do this real quick? I can do it. Alright, just hold that.
Unless you want to Bluetooth into some headphones, I can do that too.
So it's quiet.
Oh, do you want me to keep going or? So in addition to the recording, I will have this sheet. I need to make some edits before that, but it will be a workflow of everything I do today. So if you want to replicate that along with some part numbers, if you are interested in purchasing any of the items for use in your own experiments, those will be available. But again, it needs edits, so don't take what you see on the screen. The process today is going to be preparing the individual reactions. To do that, we're going to make what I like to call a sub-master mix. That sub-master mix is going to be everything except samples. I'll then aliquot that onto a plate and add the individual samples to individual wells. That reduces the number of pipetting steps, increasing our reliability. So, I have 3 samples prepared from our original stock solution, and we're going to have a 4th no template control. So I'm going to go ahead and make each of these, double checking that the assay matches what I have listed on my tube. This is provided at a 20x concentration, meaning for each individual 10 microliter reaction, we're going to use 0.5 microliters of this material. So I'm going to make enough for all of my reactions plus some extras. Okay, so we're going to have 3 samples plus 1 no template control. I'm going to run each of those in triplicate, so 3 replicates, and then I'm going to make a few extra just in case I have any pipetting errors. So I'm going to make all of that in bulk for each of my 2 reactions, and I have 2 tubes prepared here, so let's get to it. First and foremost, we have that 2x a fast advanced master mix. So here are the components for 1 reaction, which is going to be 2.5, is that right? That's not right. My math is wrong. We get to do this on the fly. Isn't that fun? So we have 5 microliters of our 2x master mix. This was originally at 20x for our assay composition, so there should be 0.5 microliters in each reaction. And then I have water that's going in. How much water? Good question. I'm going to add 2 microliters of my sample, which means I have 8 microliters for each one of my reactions. So I'm going to aliquot 8 microliters into my individual locations and then add sample to each of those, which means I have 2.5 microliters of water. I'm going to make 15 reactions total. This comes from, again, I have 3 samples. I have 1 no template control. I'm going to run each of those 3 times, and I'm going to make a couple of extra. So we run that out, and we get 15. So if we double check our math, those don't look right, so let's fix it. Always have a calculator available when you're in the lab. Okay, so this is 75, 15 times 0.5, 7.5, and 15 times 2.5 is 37.5 That should total 125. We will double check. 37 plus 7.5 plus 75 equals 120. All right. So what I'm actually going to pipette now is everything together in a bulk solution except my individual samples. It's always more accurate to pipette larger volumes than it is a whole bunch of small volumes. So this is a good approach to, number one, reduce variability and increase performance. So 75 microliters of my 2X master mix. So if you've ever used a pipette, just reminders, when you want to pick up sample, you want to make sure that the tip is fully attached. So line it up and then give it a nice solid push. You want to make sure it's dialed up for the appropriate volumes. This one can be changed. There's a bunch of different models, but you can dial in exactly what you want. So make sure that is correct. To use it, so if I'm going to pick up 75 microliters, I'm going to take that tip and depress till I get to the first stop. I can go a little bit farther if I push past the resistance. So what we're going to do is go to the first stop. That expels the air from inside the pipette. Then I'm going to slowly release that while my tip is inside the solution. That's going to allow me to pick up exactly 75 microliters. And then I move to the tube that it's going to land in, and I slowly depress that plunger, and then I go past the stop. And what that does is it takes an extra reservoir of air and it pushes the rest of the solution out of the tip so that I get everything I picked up. All right, so that was our first component. We will move next to our assay mix, which includes primer and probe. That's everything that's in here. This is for our 18S reaction. Take off the lid, and I'm going to dial up 7.5 microliters. So now I need the 10 microliter pipette that I'm going to dial up for 7.5 microliters. I have to use the tips that are designed for this pipette. I believe are these. Similar process, push the plunger down, put the tip in, pick up the solution, pull the tip out. Now go to its landing location, open up the tube, dispense the material, push the plunger, and we're done. And then we need water. We need 37.5 microliters of nuclease-free water. It is important when you're setting up these reactions, because we're working with DNA, sometimes RNA, we want to make sure we are allowing our enzyme or providing it with the right water. Yes, there's different kinds of water. There's DI water, there's nuclease-free water, but by definition, a nuclease is going to degrade DNA or RNA. We want to keep that out of our reaction or nothing's going to be accurate. So make sure you have some high quality water that you're using for the experiment. We dispense our 37.5 microliters of water. And now these are all of the primer, probe, water and enzyme, NTPs, and everything we're going to need for our individual reactions in bulk. We will then mix this and aliquot it into the plate when it's time for that, and then I'm going to add our samples to it. Next, I'm going to prepare the same for the other test. Same process in exactly the same volumes. We have 75 microliters of our 2X master mix. So we had some key points we talked about for best practices. And one of them was being efficient and clean. And some of the things you're going to notice while I'm working is that when I'm not using a tube, it stays closed. Okay, that is important because if I were to take my tips and wave them around, I can have those magical DNA particles, RNA particles, go who knows where, and we don't want that. So everything stays closed when you're not working with it. So now I can move to my assay mix. Also, keeping the box of tips closed when you're not using it will help keep your whole environment clean. It's another good tip. And now we add 37.5 of our water. Excellent. Okay, so now we are well on our way to getting this reaction completed. So for each of these, we have done 3 pipetting steps. It's not a lot, really. There's a lot more difficult processes in the lab for sure. All right, I'm going to put that to the side and talk about our plate and plate seal. So these are the 384 well plates. They are a size that allows for, it's the same size as running 4 96 samples. So 96 times 4 is 384. The individual wells of those 2 formats are different sizes. So make sure you check that table I showed you earlier about which size reactions go in which instruments, but for today and all the instruments in the bio garage, this 384 well plate is going to be what we're going to work with. So I've got a map and you'll see it in the follow-up and in my printed protocol, but what I'm going to do is I'm going to put each of these master mixes on the plate. So I'm going to put 3, A1, A2, A3, and then C1, C2, C3, and every other one going down. So I'm going to have a column of my 18S in triplicate. So one other thing I hope you notice is that when I was working with all the different components, I changed tips between each one. And that does a couple of things. Number one, it ensures that the reagents, stay clean. So I'm not taking and putting contamination into the reagents I want to keep reusing. Also, sometimes there can be liquid or other items that hang on inside the tip. So sometimes if you're doing repeated pipetting steps, they can become inaccurate if you're doing them in repetition. So I'm going to be perfectly appropriate for all of you today, and I'm going to put 8 microliters into each well, which I know everything is clear and white, and it's hard to see. So this is A1, A2, A3 with our 18S assay mix that we made. And when this is all put in row A, I will put sample 1. In row C, I will put sample 2. In row D, E, sorry, try that again. In row E, I will put sample 3. And in G, I will put our no template control. So everybody's looking at white on white, and even I am having trouble seeing what's going on. So I'm going to make some marks so we can kind of track ourselves. 1, 2, and 3, and then we will have 4, 5, 6, and 7, and then we have A, B, C, D, E, F, G, and H. That should be enough. All right. Hopefully that's a little better to see now. I'm even going to mark the edge here. Okay. So that's our field of view here. So we did A1 through 3, C1 through 3, D, E1 through 3 with our master mix. If you're pipetting and your tip touches something, you know, you just lose track of where the tip is and you happen to touch that on something, go ahead and discard it and use another one. There's plenty more tips here, and it's not worth having to repeat a whole experiment to try to save the money on one little tip. So we're almost done with the 18S addition to the plate. And then I will move on with the TERT. 18S and TERT are both really common, really good endogenous control tests. We're going to see how they compare. Against each other. Now C, 5 through 7. And yes, I'm talking to myself, and that's okay. That doesn't mean I'm insane. Maybe it does. Who knows? 6, 9, 7. Ooh, I found a mistake. Why didn't anybody tell me? I put it into F instead of E, so I'm going to recover that and move it to the right place. Excellent. All better. And now, sometimes you can tilt the plate like this and get a reflection off of the solution surface if you're having trouble seeing what's going on. G5, G6, and last one, G7. All right. The rest of this can be discarded. All right, before I move on, I'm going to take care of my items and put them back on ice so that they remain useful for the next experiment. Now what's left? Putting sample into that reaction mixture. So the way this was set up is I had 8 microliters of my master mix, and now I'm going to add 2 microliters of my sample to each one of the reactions. It's nice to have your workspace organized and easy to use. I like my tips, my waste container on my right because I'm right-handed. So think ahead about how you're going to be working in this space and the things you need before you start. So to each reaction, I'm going to add 2 microliters of sample. I have 3 samples labeled sample 1. I know, creative. Sample 2 and sample 3, and then I'm going to add 1 of a no template control. So remember, I added the 18S reaction mix to columns 1, 2, 3, and I added the TERT to 5, 6, 7, and now I'm going to come back and add sample 1 in row A, sample 2 in C, sample 3 in E, and my no template control in G. So if we work out the rest of that metric, I'll have triplicates represented for each of the 2 tests for each of the 4 samples. So I'm adding 2 microliters of sample to each reaction. Why 2 microliters? Well, I already know what the appropriate concentration should be. Why 2 and not 1? Well, that's an excellent question. Let's talk about it. 2 microliters is more accurate to pipette than 1 microliter. And I don't know how you guys do about calibrating your pipettes in your labs, but I was never very good about it. So if there's anything wrong with your pipetter, you're going to have more problems at 1 microliter than you will have at 2. So I like to recommend 2 microliters just to be consistent and accurate. Okay, so that was sample 1. Now in row C, I'm going to add sample 2. Changing tips between each one to make sure I'm adding the appropriate volume. I'm also using my finger to track where I'm at. Some other useful techniques are to coordinate the tips you're using to correspond to the plate format. That way you know if I used that tip, I completed the sample addition. So that was C, D, E, 1. And 3, skipping 4, and then we're adding 2, 4, 5, and 6. When you are pipetting, I don't recommend trying to carry on a conversation with anybody else. It's really hard, and you lose track. So sometimes you just have to say, not right now, friend. We'll talk later. So now I'm preparing my no template control. So what is the purpose of a no template control? It is to assess how your process is viewed without template present. So if your assay is not performing well, you can see extra amplification in those no template controls. Also, if you have process issues or contaminated reagents, you'll see them again in those no template controls. So please always use those. They are important. Don't skip them. It's really not worth the savings if you have to go back and do 4, 5, 6 experiments because you simply skipped your no template control. I did write on the top of the plate so we could track what was being added. Don't write on the surface. Remember, we're going to image right through the top of those openings that we just pipetted into. So you don't want any marker there. They will fluoresce. Marker fluoresces. I know, I didn't, you know, the things you learn. All right, but we do need to seal this because we only have 10 microliters in each of these locations. So we do need to seal it. This is the box of covers. They are called micro-optical adhesive films. And it's the optical part that is really important. There are going to be other kinds of films that may be around. Just make sure it says optical. So these are made in exactly the right size to fit on top of the plate with some extra for you to hold on to. So if I separate this, I can take off the backing. I'm going to hold on these end tabs. And it's going to be kind of hard to see, but each one of those, right where it's perforated. So what I'm going to do is I'm just going to line that up as best I can. It's not worth getting really stressed out about. I have a paddle, and what I'm going to do is I'm going to go from the center out. Both directions, and I'm going to do it again horizontally. All right, should be pretty well stuck. I'm going to take the fins off. It's perforated, so just snap those off. Okay. Now what I want to do is double check that I actually have a good seal. So I'm just going to go back over the locations that have samples in them. Give them a good press. And sometimes I'll even take a gloved hand and run my finger over those just to double check that that seal's completed. You're going to have a really hard time seeing this on camera, but often what you can see is if there's an incomplete seal, you'll actually see a difference in color. So it almost looks, the reflection is different. So if it's not stuck, it'll actually look like a white spot rather than a nice dark circle. Okay, one of the questions that came up in the talk was about vortexing. So now I'm going to hand the plate off to Molly to go vortex and spin down before we start. And we'll move to the instrument to go over setting up the run. So at the computer, we have two components. We have the computer that is doing the controlling. This is where we're going to set up the plate, set up the run. And then we're going to move to one of the accessory instruments. This is where we're going to actually initiate the run and load our plate. Loading a plate, there's an eject button here at the top. That is the only way to access that block. I push the eject button and that drawer will come out. It's waking up. It went to sleep, so it's waking up. And when it does, that will come out and we will have access to the block where the amplification is going to happen. Okay, you can see this is the same format we just pipetted into, and we're just going to drop that plate right in when we're ready to go. Going back to our software, when Molly's back, we'll start that. But let's get set up to actually complete the run. When we launch the software and we're brand new, we want to start with setting up a plate. So what we're going to do is click Set Up Plate, and next we want to be as specific as possible. Going down the left-hand side of this column, you're going to choose your instrument. The BioGarage has the Quant Studio 7 Pro. We are using the 384 well plates and blocks. We want to make sure that is selected. We are going to run, we had a fast advanced master mix, and it happens to run in fast mode. Our enzyme is super speedy, so we can run fast. And then our experiment type is relative quantification. Once we've made these selections down the left side, what it does is filter our available preset protocols down to a really reasonable list of 2. What we're left with is one that says comparative CT and one says comparative CT with MELT. Just for nomenclature purposes, TACMAN assays are not useful with MELT, so we're going to use the one without MELT. So what this does is we have 3 tabs at the top, and what we've done is loaded you a template that is going to make the most sense. It's always recommended to check with the parameters of your specific assay and the parameters associated with your master mix and enzyme to make sure that we've prepared. If we are going to run a reverse transcription reaction, I need to add that upfront 50-degree step. So I'll do that quickly, add, hold, and we will run this at 50 degrees for 5 minutes. Again, check with the manufacturer and user guide for those items. Next, we move into the enzyme activation step. We've tried to preload these default templates with the information from our preferred products. So this is already defined to go from 95 to 60. It's going to do that for 40 cycles with an enzyme activation step ahead of time. Just going to call out here, we have a camera here. That means this is where we're going to collect all of those data points when we move through. Last thing to make note of on this page, and it is important, is right up here where it says reaction volume. We ran a 10 microliter reaction, so we're going to modify that. And now this page is complete. We go to our setup, and I'm going to add 4 samples. One of them was my NTC, and I have 2 targets, and I'm going to do this quickly, but you can customize this as much or as little as you want. You can also make changes after the data is collected. So I'm going to show you one, and then I'm going to do the rest when the run is finished. So I have added 2 tests to the plate, and now I'm going to add 2 samples using the checkboxes. All right, so we can go through and fill the rest of this out after the run is done. So one other point to highlight here is this window where it says rocks. Check with the manufacturer's information about your master mix. We use rocks as a passive reference normalization, and I highly recommend it, but it's not in all master mixes, and it's your choice to turn it off or leave it on. All right, so once we're happy with the plate setup, we can move to the run summary. From here, it will highlight our amplification protocol. This is the last step to double check everything. It's going to show you what you added for the plate map, which we didn't finish, but that's okay. We keep scrolling down, and now this is where we have some actions. Now it asks you if you want to save that plate protocol. Right now, I'm going to go ahead and send this to instrument 1, which is how I connected, so double check the name of the instrument, and I'm going to send it to run queue. So it asks you if you want to save it, how you want to save it, and yes, I'm fine with that. Okay, so green check mark, we sent the file from the computer to the instrument. Click done, and now we're finished with the computer. Going back to the instrument, I'm putting the plate in, always double checking that A1 is in the upper right-hand corner. So A1 named on the plate, it's also molded into the surface of the reaction. Make sure it seats on there so that the skirting is flush with the plastic holder for the block. and then we can close that. All right, there's a great big button in the middle of this screen, and that's what we're going to use. We click Setup Run. And now, remember on the local computer, I sent it to the Run queue. So now I select Run queue. And here is that template, which I had untitled. You can always check those names, and they do match. So now it's going to ask you where you want to save it and what you want to do. Again, this is up to you to decide how you want to configure it. You also have a last-minute option to change your protocol or your plate method. But when you are happy, you can click Start Run. Did you remember to insert the plate? Yes, I did. Start Run. And now it will start. Here it's green when it's loading, and then when it starts amplifying, it will turn orange, and it will give you a countdown clock to a completed process. Okay, so we will wait for that to finish, and we will come back after lunch and look at some data analysis. Thanks everybody, and I'll be downstairs later if you have any more questions.
So this is another copy of the software I have right on my computer, but you will have access to this same software downstairs in the bio garage. So when the run is finished, we can come back, and we first looked at setup plate, but now we're going to view the data. So let's click on view data, and it's going to ask you to find that file. If you can't find it in the short list, you can always open file in a new window and navigate to it. So this is where I saved that file. I should have named it. Sorry, I didn't. So that will open this window. So I went ahead and I filled out the rest of the plate setup. Zoom out a little bit. So just a reminder, we had columns 1, 2, and 3 as replicate samples. Same 5, 6, and 7 were replicate samples. We put the 18S in columns 1, 2, and 3, and then 5, 6, and 7 are as the TERT assay. Sample 1 in row A, sample 2 in row C, sample E, or sample 3 in row E, and our negative controls in G. So from here, so this is just the labeling convention. Under run summary, what you get is a lot of information no one ever really looks at, except maybe someone like Molly, that tells you when everything was calibrated and when it's time to do that again. But it was run, the run protocol completed. So it finished, time, start, and stop. The first step when you actually start looking at data analysis is this quality check tab. So this is a really great overview of your data with the purpose of making sure all of the data you collected is worthwhile inputting into the data analysis. So we're going to go ahead and give it an overview and check that we're going to use it. So how do we do that? There's a lot of great ways to do this, but in summary, this is already in a logarithmic format. Remember, we talked about log versus linear. So the software also, because by default, it will automatically assign a threshold based on the data that it's presented. Caveat to that is if you assign the whole plate with a sample name or target name, it will go ahead and use all of those wells for that assessment. So if you only have 5 samples in a plate, you now have most of the plate being erroneous data, and you'll likely get some weird reports back. So just make sure that the assignments are only for sample containing wells. Okay. From there, these interactive, this is an interactive plate map. So as we move through and select different wells, it will highlight in the table and also highlight the amplification plot we're looking at. We do have a yellow triangle, and if you hover on this, it will give you information about why we have those yellow triangles. And most importantly, I haven't completed the assignments to actually do the gene expression calculations, and we'll do that together. So to assess the quality of the samples, remember we're looking at good quality amplification, nice and strong. Let's expand this just so we can see it a little better. And we had two assays. One was the control assay and one was the gene of interest, and they are significantly different in expression levels. We can sort these by looking at individual assays. So our 18S was a good, strong performer, and our TERT was less strong, so a lower expressor. Some other things I would want to look at. I would want to look at my negative controls to make sure they're not amplifying much. So it looks like I may have one erroneous 18S amplification, but it's pretty high in concentration. So I'm going to go ahead and give myself an okay. I won't give myself a great, but I'll give myself an okay. We all do stuff. If we knew the answers and we were all great, we wouldn't run experiments. Okay. So we can go ahead and move to the next step, which was we've established that this plate is worth looking at. We have good assays. Our negative control is okay. But what do we do next? Next, I would want to look at the quality of the actual CT values I generated and the quality of those amplification curves. So I'm going to minimize this just a little bit so we can see more of the table. And you can see there's a lot of data here, but if you keep scrolling over, you're going to start to see numbers like CQ, CQ confidence, AMP score, AMP status, and a bunch of information. So let's take a stop here at CQ confidence. So what we are looking at here with this value, it's not a reportable number, but what it does is it's associated with the quality of that CQ value. We can use it to help delineate, do I want to accept this or not? And let's look at the amplification curves. So I have a bunch that say undetermined for CQ, and the CQ confidence is 0. Those are my negative controls, and they did not amplify. So that's pretty clear. But then we may have others that have lower quality, and unfortunately, this data is all too good. So I would say anything that is below 0.6 for a CQ confidence is probably not worth keeping in your data. That's the curve quality and the CQ it's assigned. The software doesn't have a lot of confidence in that data point. So either it's inconsistent, or the shape of the curve is wrong, or it just doesn't look good. So that doesn't mean you have to throw it out, but I think you need to evaluate that data with some caution. All right, so anything between 0.8 and 0.6, I would put in that marginal range and say, let's look really hard at this. Look at technical replicates, look at the shape and consistency of the assay as a whole, and move from there. This is all about what data you want to put in to your analysis. Doesn't really matter my opinions. It's your data, and you need to stand behind it. If it were my own personal experiment with an assay that is well qualified, and say a TACMAN assay I purchased from Thermo Fisher, I would say anything below 0.8 I wouldn't even consider. But that is my personal opinion and not a rule. Just make that clear. All right, so those are also associated, CQ confidence and then AMP score and AMP status. Those two are tied together and related in a lot of ways to that CQ confidence, but it's more associated with the shape of the curve that it is actually appropriately growing in that twofold manner. So again, I have an undetermined CT with a CQ confidence of 0 and AMP score of 0.5. So this is on a 0 to 1.5 scale. And then it says no AMP. So clearly that is not a good data point, but it's my negative control. So it's actually a good data point. All right. That brings me to a really interesting point, is back in the run summary, the plate setup, I assigned all of these as unknown samples. which is fine, totally fine. But this assignment does have an impact on how it's flagged and utilized. If I then took this and changed it to a negative control, I get the spinning wheel, it does things. Now I need to reanalyze. Yep. Okay, so back into quality check, it now has different metrics. Now it says task negative control, and my metrics now appear pretty positive. A lot better. Okay, so that's how you do it. It also would throw flags if it sees a negative control and a lot of amplification. It can potentially throw those flags. And those flags really are points for attention. There are no rules that if you have a flag, it needs to be thrown away. But it is a place to focus your attention. Do I have a high standard deviation of technical replicates? If you do, you might want to consider throwing one of those away. If you have, low CQ confidence, that can throw a flag as well. Those are customizable under the primary analysis settings. So here under QC alerts, all kinds of things that you can flag for your results. So lots and lots and lots of options. But for the most part, all of this data looks possibly even too good. But hey, not gonna brag. All right. Before I move on, I wanted to point out each of these assays, each of these tests is going to be evaluated with a threshold value independently. They are separate tests. They are going to be assessed individually because they have different properties. They have different expression levels. They have different efficiencies. So it doesn't make sense to lump all of the data together, but more separately and individually. All right, so ultimately, all of this data we're going to keep. If you want to exclude some, that's pretty straightforward. You can omit them and then reinitiate the analysis. Straightforward. All right, what's next? The whole point of this was to do gene expression, which is under this tab called relative quantification. So let's click on it. Oh no, there's nothing in my graph. That's okay because I haven't told the software what I'm doing. Remember, there were the 4 components of our gene expression experiment. We had a control gene. We had a gene of interest. Those are 2 different tests. Then had a sample for reference and a sample of interest. We haven't assigned any of that. So let's do that. That happens up here under Actions, Relative Quantification Analysis Settings. So I'm going to tell it how to do stuff. All right. So first one, endogenous control. I'm going to pick 18S as my endogenous control, and then my TERT gene as my gene of interest. So I just check this box, and I can apply that. Next. Oh, we're getting somewhere. My table changed. Okay. At the same time or separately, we can now assign which sample is going to be my reference sample. I'm going to pick number 1. I don't have a preference. We can change our mind later. That's the lovely thing about all of this data is whatever you're doing, it saves the raw data in the file. You can do all kinds of different analysis. You can even, to the point where, recover data from unassigned wells. You're going to collect all the data across the whole plate, and you can change whatever kind of analysis you input on that at any point in time. So let's apply this. And now we get some plots. Okay, so what do we see? We see that we have our 2 targets. We have an EQ-CQ mean. That's a lot of letters. That's essentially the mean of the CQ value. And then we have an adjusted EQ-CQ mean. We have a delta EQ-CQ mean. We have essentially delta with the mean, delta with the standard error, delta delta CQ, and then RQ, RQ min, and RQ max. These four here are the numbers that actually get input into this plot. Okay. What happens if I change my reference sample? Well, in this group, not a whole lot because I sort of faked it. No, I didn't fake it. I don't fake anything. They're actually all from the same sample in different dilutions. So let's change this to sample 3. Apply. And we're going to get the same result because it's the same data applied in a different way. So these were different dilutions of the same sample. So our data is showing exactly what we expected, is that there is no change. There is no change in the relative expression of these samples because we're comparing what's going on inside the sample and not the quantity of the sample. We changed the total quantity we put in, but the relative change is the same between the two genes that we're looking at. Okay. This is a little easier to understand, so I'm going to open an example file. I think that'll be easier to understand because there will be variation. So let's open. I want my examples. So when you download this software, you'll get some example files, which I think is great. And let's choose this one. All right, so this has some more changes. We can do a lot of the same things reviewing the data. This is an example file, so all the data is pretty great. So now when we go to this relative quantification table, we're going to start seeing some differences. So what samples do we actually have on this plate? They are named 400, 800, 1600, 3200, and 6400. We have IPC, which is our control, and we have RNase-P, which is our gene of interest. Those have already been assigned in these groups. So when we go to our relative quantification example file here, we can see we've got our error bars presented. We have RQ value on the y-axis. And now we can see in our data table that our 1600 sample is our reference. And so our RQ is 1. By definition, your reference sample RQ is always 1. If we look at 1600 versus 3200, we have an RQ of 1.9. And that kind of makes sense because 3200 is twice 1600. So maybe the quantities are the names, just guessing. I don't know that for sure, but it seems logical. And then if we double that again, it gets 6400. We now have a value of 3.6. So we're in line with potentially some additional variation as we change concentration. All right. So this is what we're looking at. If at any time you want to change, we can see how these numbers change. Relative quantification analysis settings, we can change this to say 400. And now our RQ values, the difference between the size of the bars remains the same, but the RQs are now different. So now we have an RQ of 1 for 400. And then our 3200 is now all the way up to 6.8. So this is how you go about looking at the data in gene expression. So again, the hard part's actually getting the information into the software, not necessarily getting plots out of the software. And honestly, the most physically demanding is setting up the plates. But is it really that hard? And you can always ask Molly to do it for you. So, and then intellectually picking what should take the most time, in my mind, what should take the most time is picking the assays and doing your upfront work to make sure you are getting high quality data before you even set foot in the lab. There is no excuse in my mind for that. You have all the time in the world and it only costs your time, which I know to me is really valuable, but to your PI, maybe not so much. So all it takes is time. to do that. And if you spend the time doing it, you will set yourself up for better data in the end. Better data leads to publications, and that's what everybody wants. So take the time to make sure you know what you're doing, where you're going, and then go get it. All right? Questions about data analysis? Personal preference, I had a lot of room on the plate, and just to avoid any cross-contamination issues, I did. If I'd have had more samples, I would have not done that. Sometimes if you have, say, an 8-column multi-channel pipette, you'll go every other one just because that's how it fits into a 384-well plate. So no good reason. Sorry. Yeah. Right. I don't. We can look at it. But the big difference, and hopefully I mentioned this at some point today, but when you're working with TACMAN assays, what you're doing is you're actually going to degrade that probe to generate your fluorescence signal. So that is inherent. We're not going to be able to go back and reassemble that probe and turn off the signal so we can turn it on, which is really important for a melt curve. So typically you're only going to see melt curves as part of a cyber type reaction, which is a reversible signal. When you have double-stranded DNA, it sticks. If you heat it up and you denature that DNA and make it single-stranded simply by heat, the cyber molecule will fall off and you will get no signal, which is what we take advantage of in a melt curve. So let's look at a melt curve. Yeah. Who was that? Yeah. So while I look for, let's get out of some other ones. Need more options, Where am I with melt? I know I have with melt. Or just melt? Huh? Oh yeah, there we go. Curve. Yes. Yeah, this is a tricky subject. I can get into the weeds a little bit if I talk about my personal preference, but just from some pitfalls I've seen people fall into in the past, I'm definitely going to recommend TACMAN, and those inherently don't have melt curves. I have this data, I just need to find it. Desktop. Yeah, and like I said, the PI doesn't always value our time, but I value my time. So if we can work with you on price and get you on board and getting more data in less amount of time, we're going to help you. But some people are really stuck, and we're stuck on whatever we have. So let's talk about how to, I'm sorry, this is taking me so long. I don't want the D drive, I want the C. Yeah. Okay, so this is another software. I just couldn't find the file in the other software, but they're cross-compatible. But essentially what we're doing, just to orient you about what we're actually talking about, is we're going to run a standard amplification protocol. And then when we're done, so remember with cyber, all of that QPCR data we just collected is a lump sum of all the reporters. And with that, you want to make sure you made what you thought you made. Okay. And one of the checks we can do on that is running a melt curve. And that melt curve can give you information about the width of that melt curve can tell you about the amount of side products, or additional peaks can tell you information about specific side products. So there's definitely some metrics that we can take out of the melt curve and apply to the quality of our data. So definitely during the development stage, I highly recommend a melt curve. But what we're going to do is we are going to finish our amplification, collect all that QPCR data, and then we're going to heat it up and we're going to cool it down. So we're going to denature everything until it's single-stranded, and we're going to make it cool. So cooler than our extension temperature, so at least equal. And then we're going to start increasing the temperature again to melt it apart slowly. So remember, what we're doing is we're starting with high signal, where everything is bound together in double strands and producing signal. And then we're slowly going to increase that temperature and consistently measure the fluorescence as we go, so that we watch it slowly fall apart. We will eventually get to the point where everything falls apart in a big reaction. So it's going to be a slow decrease over time. And then eventually we get to this point where it kind of just gives up and says, ah, forget it, I'm going to fall apart. Well, we call that the melting temperature. It's where that happens in a big way. So we can look at this melt curve plot. This is what people put in papers, look at regularly, but you can look at it instead of in this derivative plot, in the normalized plot, which is what I just walked through conceptually. We start at a low temperature, so high fluorescence, and we walk through increasing that temperature until we get to that TM point where the signal just drops off. That's where everything just, I'm done. And then we get to a very low point of fluorescence where everything is single-stranded. It's hard to determine that TM accurately in this linear plot. So we take the first derivative to figure out where the maximum change happens, and that is here. Normalized to derivative plot. And now we get that nice peak. When you're looking at melt curve data, you want the S's. It's a single, smooth, sharp, symmetrical peak, or 4 S's. So that's what we're looking for. So obviously this was a really successful melt curve. But again, it's only applicable to cyber reactions, not TACMAN reactions. And I'm happy to go through this in the other software as well. I just was struggling with data files. So happy to show you how to do that another time. Other questions? You're all pros for, oh yeah, go ahead. Yeah, so you actually have to write across the well. So the way that instrument is set up is we focus the cameras and isolate just the wells. So we only actually capture the fluorescence inside of that defined well area. So as long as you're writing outside of that, you're fine, just not inside. So if you were to take a Sharpie and go across the plate surface, we would see that. And I have an example of that if you want to see it in another presentation. But yeah, just if you need to write on the plate, which everybody wants to write on the plate, I do it, other people do it, just make sure it's on the edges or away from the actual well itself, or it can bleed in.